tell us a little bit about your background and the laboratory and the clinical work that you do.
Actually, I'm my major is in biology and chemistry, so that's where I started out, and then I decided to go to human genetics, and since almost 30 years now, I'm working in human genetics, and my focus in human genetics is eye disorders, and specifically retinal degeneration. So my interest in human genetics is the genetic variation in humans, and what that genetic variation can do either to keep you healthy or actually to lead to disease.
So tell us something about the presentations that you made here at Arbor 2016.
So I had several posters here and several poster sessions where I actively participated. And I think it was really good to sort of get feedback from my colleagues and also to discuss certain topics. Basically, what we do is we look into genotype-phenotype correlations. That's one part. So we want to understand the various clinical phenotypes based on the genetic component involved. At the same time, we are interested in translational research. So we want to bring some of our work actually into clinic. So we are very much interested to help patients, and so this is particularly true for a disease called Best vitelliform macular dystrophy. Another disease is the X-linked juvenile retinoschisis. It's an X-linked disease and young men are affected. So we have certain things going on right now, and we try to push it ahead to really get into clinical trials. I think a trend we see all over, which is a good situation in 2016.
So can you summarize the results of the material that you presented?
I mean, we are interested in novel disease genes, so meaning we are recruiting families where there is a disease in a Mendelian fashion, meaning it's either passed on in an autosomal dominant or recessive or X-linked fashion. And then we try to identify this gene, we try to identify mutations in this gene, and then we learn more about the function of these genes, proteins. So a major part of my laboratory is actually interested in understanding functional differences. And another major part is interested not only in these so-called Mendelian diseases, but in the complex diseases. So another big emphasis in my laboratory is basically age-related macular degeneration. So here we try to understand the genetic architecture, so the entire genetic components contributing to this disease, and then to definitely get ideas how we could find some maybe novel treatment options. So this is major part of my work, and this is all part of what I presented here.
In terms of milestones in your work, could you tell us a little bit about the future trajectory?
Yeah, as I mentioned already, my big goal is to really translate basic research into clinical research. Which is not always that easy, and which usually takes quite a bit of time. So what we have done we have provided several examples in my research, where we started out with identifying the gene, then tried to understand the function of this gene, pathways, and then sort of developed treatment options, tested that in animal models, very often in mouse models. And nowadays we are also very much working into induced pluripotent stem cells. So this is a novel technology, a few years old now, but it's a very powerful technology because you derive sort of cells, skin material from the patient, you cultivate that in the dish, and basically you can do then mechanistical experiments on these cells, but also you can test, for example, medication on those cells. So in one project, what we are doing is we screen basically such cells on chemical compound libraries to see whether some of the deleterious effects of the mutation can be ameliorated by sort of certain chemical compounds. If we are able to find one such compound, then we can sort of suggest that as a possible treatment option.
If you were to tell patients what the anticipated benefits of your line of research would be for them, what would that be?
Yeah, there are several advantages coming out of the kind of research we are doing. One definitely is that we are doing DNA testing. In Germany alone, there are patients usually waiting on average for about 3 years for their proper diagnosis. So especially when it comes to rare diseases. And so DNA testing is actually a major and important step to really provide sort of the diagnosis for the patient and also to provide the proper diagnosis for the patient. Very often, as you know, in retinal diseases, there is a phenotype, there is one single phenotype, but that doesn't mean, so there's a lot of underlying causes. And finally, to sort of offer the patient a proper treatment, you need to know exactly what the underlying causes are. And that's basically what we, so that's on that part of diagnostics. And of course, we also want to understand the pathways then in disease, so to provide the patient with an explanation what went wrong in his eyes. And as I said, sort of the next and final step would be then to really try to individually address these changes and maybe to make them somehow better.
Professor, is there anything else you'd like to add about your work and about the future of this particular area of research?
I have to say I'm 30 years in the field now, and when I started out, we were at the very beginning, and we started out get to know a little bit of genomics and genes, and and now we are at a situation, and specifically ARVO 2016 was an excellent example, so that we have made major advances in this field, and now there is sort of hope for the patient to actually receive a treatment nowadays.